And welcome back. In recent news, Einstein research demonstrated the successful suppression of HIV infections in mice. The scientists developed and used a novel synthetic protein to boost the immune response, and the technology is being studied for head and neck cancers, type 1 diabetes, and other autoimmune diseases that could possibly offer a path to a functional cure for HIV and other chronic viral infections. Joining us and sharing more details is the director of the Einstein Macromolecular Therapeutics Development Facility and chair of biochemistry at Einstein, Stephen Almo, and Charles Michael, chair in autoimmune diseases at Einstein and director of Einstein Rockefeller CUNY Center for AIDS Research, Harris Goldstein. And both of you, thank you so much for joining us this morning.
Our pleasure.
Thank you.
Thank you. And I guess, I want to talk a little bit about this treatment strategy that's actually used to possibly cure HIV. It looks like a great breakthrough here.
Right. Well, I think that really have to think in terms of how you conduct the immune response. And our body really does a pretty good job of developing an immune response that in most cases is able to very effectively control infection. However, HIV is one infection, unfortunately, that our body can control, but it can't eradicate. And part of that is that the HIV distracts the immune system into targeting a response against areas of HIV that it could readily mutate and thus evade the immune response. And what this product, this Syntac, that Dr. Almo and his group have developed that we now are applying to HIV allows us to do is really potentially give to patients and focus their immune response against the Achilles heel of HIV. And that really is to us the breakthrough. Now, in terms of curing HIV, I think it's unlikely that we're ever gonna, it's unlikely right now that we can eliminate every last HIV virus in individuals that are infected. However, if we could teach their immune system to suppress HIV infection so that as soon as it emerges, kind of like a sprinkler system immediately puts out a fire, then we could potentially provide what we call a functional cure. Namely, their immune system is controlling the infection the same way the drugs right now can control the infection, but the patients don't have to take drugs.
Yeah. And so, Dr. Almo, a little bit more about this novel synthetic protein that's called Syntac. Talk to us about your work and how it works.
Right. So I would start with a simple explanation. Everybody's had a blood test taken. And when you have a drop of blood, it's red because you've got red blood cells. A very small fraction of those cells are actually immune cells. And those are really the gatekeepers of health. They fight against autoimmune disease, infectious disease, malignancies. And those cells specifically recognize unique features on the diseased cell tissue, either an infected cell or malignant cell. And that address that they recognize, if you will, allows them to deliver various signals that they can either eliminate those cancerous cells or cause other cell types to expand and enhance. So what we've tried to do with our Syntac technology, these proteins we've made synthetically, is to mimic that process. We really, on our proteins, we've taken features that are present in these immune cells, encoded them in our proteins so that we can specifically deliver to those immune cells activating signals. And in this case, that activating signal specifically turns on immune cells that are encoded to go after and hopefully delete and kill HIV viral infected cells. So it's really like a mail delivery process. There's the address, the contents of the envelope, if you will, and then the delivery process itself.
So share me a little bit more, because I'm interested in knowing how this actually works between the mice and the humans, because there's a correlation right there. So can you make it a little bit more plain for our viewers?
Right. So despite the fact we look somewhat different from mice, we also have a lot of commonalities, shared features in our immune response. And what we were able to do was generate molecules that work both on human immune cells as well as mouse immune cells. But what Harris has done, and I think what I'll let him describe in a little more detail, he's developed what he calls a humanized mouse system that allows us to study human cells in a living organism that is the surrogate mouse. So Harris?
Right, so it raises a really important point, namely what works in mice may or may not work in humans, clearly. So in order to overcome that, what we have done is that we have developed a a humanized mouse model where we basically take these white blood cells that Dr. Almo discussed coming from a patient, and in fact, coming from an HIV infected patient, we inject them into mice that really have no functioning immune system. Because these mice don't have functioning immune system, they will not reject the human cells, and the human cells will continue to proliferate and act inside the mouse. And because it's coming from an HIV infected individual, then HIV, because the mice are not on any medication, can now come out and start infecting cells and developing a very significant HIV infection. This now allows us to treat these mice with these syntax to see if we can now amplify and expand the very focused immune response that T cells can give to control and eliminate HIV infected cells. Can we suppress that infection? And in fact, in this paper, we demonstrated that we can. So in essence, we've shown it in actual human cells taken from an HIV infected patient, which really makes us optimistic that this may actually work when we give it to patients.
So we hear a little bit about antiretroviral therapy. How does this protein actually compare to antiretroviral therapy?
So antiretroviral therapy is very effective in patients, but patients need to take it constantly, every day. They need to take it. If they stop taking the medication, then the HIV that's been basically lying quiescent, silent in the body, reemerges because it no longer is being suppressed by the medication, and the patient now will have a full disseminated spread infection of HIV. What this particular syntax does is it basically expands immune response that could potentially control and eliminate HIV infected cells as they emerge. So it's basically give the medication, expand the patient's immune system, and then from there, the patient's immune system takes over, and the patients no longer would need to take medication every day.
That's
I
awesome.
would just add to that, so they will stay around and remember what their job is, supposed to target and eliminate these HIV infected T cells. So this is something that the retroviral therapy does not have, hence you have to take those small molecule drugs every day. Once you establish this immune sort of protection, it can last for a very long time.
You know, and I would compare this mechanism to how a vaccine works. So in essence, you vaccinate a patient, you now expand the immune response that's able now to control or prevent infection. The advantage of the syntax over a classic vaccine is where a classic vaccine will activate a wide array of immune cells, many of which, in the almost overwhelming majority, cannot control HIV infection. Because we know what kind of cells can control HIV infection, we could focally expand those specific cells and now enable them to control the infection. So the patient's own immune system now is able to take over and control the infection and take over the job that the antiretroviral therapy was doing beforehand.
And before I let you go, is there a timetable between the time of discovery and the possibility of actual treatment?
So I would say that's very hard to judge. If I had to guess, I would say it's really in the 5 or longer year timescale, because again, there's a lot of work that needs to be done. We've done some very elegant basic science work. We now need to try to translate into the clinic. The one thing working in our favor, though, is that somewhat different versions of our syntax drugs are already in clinical trials for treating head and neck cancer. So they've already been in upwards of 50 patients and have a very good toxicity profile, meaning that they're really relatively benign after application. And these, like I said, are in clinical trials for head and neck cancer in a number of settings. So that's really going to boost or shorten the time scale, I should say, for which we might be able to achieve a clinical outcome. I should also add that these clinical trials are in collaboration with Q Biopharma, of which I'm a founding founder. And Harris and I have both previously received research funds for a different project from Q for full transparency.
Certainly a lot of information there we will continue to follow. Thank you so much to Dr. Stephen Almo and then also Dr. Harris Goldstein for being with us here on Open. Now, if you want more information, we encourage you, visit the website at einsteinmed.edu and then also follow them on Twitter and Instagram at EinsteinMed. We do have more shows, so we encourage you, don't go anywhere. Open continues coming up right after this.